Good morning, everybody. Welcome to day 4 of Bioinformatics. I'm so glad you're back. I see that some folks have arrived just now. That's great. Welcome in. I'm glad you're here. We are going to spend some time talking about protein structure today, because I really think that we don't emphasize enough how the structures taken on by biological molecules affect what they can do, what activities they have. So today, we'll have this discussion of protein structure and function. Now, I've altered the slides a little bit from the version that was posted yesterday. One of the things I realized was that I'd never updated my table of contents. So we start with this idea that structure is the basis for function. So we're going to talk a lot about that in two kind of canonical ways in which structure changes to reflect function. The Tanford Transition and the Catalytic Triad. Both of these have been studied to death, and there's a pile of data about them. We have good stories to tell. From there, we'll move on to a discussion of protein structure from a perspective on how we can experimentally determine what structure a protein adopts. We'll discuss phi and psi angles along with Levinthal's paradox, kind of an interesting kind of personality-driven way to look at what we've learned about how structure comes on. And then we're going to move into some really amazing bioinformatic methods for de novo prediction of protein structure. Does anyone know what de novo means? Creating something that's brand new, right? So if you don't have any structural information about a protein, you have only its sequence, de novo algorithms will attempt to tell you what the structure of the thing is, starting from sequence alone. That is a very bold challenge. And then we'll talk about a competition of sorts that's been running for many years in this field called CASP, and the kind of evaluations that have come out of CASP in recent years that shows you sort of the modern trends of thinking within bioinformatics. Okay, now we're going to start with a little diagram that could easily have been drawn in the late 1950s, because a major paper published, I believe, in 1952 spelled out this 4-level structure of proteins. Does everyone remember a diagram something like this from your biochemistry class? Okay, some yeses and some nos. All right, well, we'll just walk through it. Typically speaking, we always think of the way that amino acids are physically connected to each other through covalent bonds as the primary structure of that protein. So if you look at the sequence, it's just a bunch of letters, right? But each of those represents an amino acid residue that's connected by peptide bonds to the next amino acids, the one before and the one after, right? So that amino acid structure, the amino acid sequence connected through peptide bonds, constitutes the primary structure. At the next level, we have a lot of things that are happening because of interactions among different elements of the backbones and of the amino acids. And for that, we get secondary structure, alpha helices and beta sheets. So an alpha helix is pretty much a helix, right? You have a series of amino acids through which the backbone forms this helical structure. It's stabilized in that way, and it acts like these kind of rigid poles of structure within the proteins. Beta sheets can be a little different. You can have parallel structures where they're running N to C terminus. Remember, we have one side of the amino acid that's a nitrogen, the other side that's a carboxyl. That's the N and C termini. So we can think of things running parallel, or we can think of things running anti-parallel. These things can form sheets then of different chains of amino acids that are all formed that are all stably in structures next to each other. Now in tertiary structure, we have kind of a higher level form taking on. That this entire polypeptide is going to fold up in some way that has some canonical fold, where these little domains have all packed against each other in some sort of reliable way to form this tertiary structure. Now at this point, we often think about cysteine, which in my mind is one of the weirdest of all the amino acids. What's weird about cysteine? There are 2 amino acids that incorporate an element found within cysteine.
Sulfur.
Sulfur, very good. Yes, sulfur and methionine, which are both, and frankly, very strange amino acids. The cysteine really tops them all. The cysteine's got this sulfhydryl group in its side chain. And when you have 2 cysteines near each other, they can form a disulfide bond. So for you, the term disulfide bond might come with all kinds of meaning and stuff. But for me, whenever I see a pair of cysteines together in a protein structure, I think of a safety pin, right? You're putting together some sort of unwieldy Halloween costume, and you need this little bit to stay attached to that little bit. You're not going to take all the time to stitch it together, but you probably have a safety pin and you can attach it there. In the same way, the protein structures are going to use these disulfides as safety pins to kind of hold that fold in place. All right, so that's tertiary structure. And finally, you may have multiple polypeptides that are all interacting to form one large complex. And for that, we would say we have quaternary structure. Furthermore, many protein complexes have elements that are not polypeptide at all. For example, hemoglobin, right? What's the difference between hemoglobin and myohemoglobin?
The one is like one molecule and the other one is, I think, 4 back together.
Okay, so it is true that hemoglobin is 4 polypeptides brought together, I think 2 copies of 2 different ones. But the other difference is that it contains heme.
Oh.
Heme is the iron-containing element, right? So iron is not part of any of the amino acids, but that element gets added in as part of quaternary structure. So we think of all 4 of these levels as representing the canonical 4 levels of protein structure, as stated in the 1950s. However, in doing a little bit of research this morning, sometimes I just get curious, I'm sorry, I realized that modern protein structure people are starting to think on a much more finely grained level about protein structure. So at the bottom, we still have our linear sequence of amino acids. We still have these connections forming between pairs of amino acids through peptide bonds, of course. From there, we have units of secondary structure that are largely just self-forming. If you produce any sequence of this, whether you've got chaperonins or whatever present in your mixture, these parts of the sequence just fold up this way because it's energetically favorable for them to do so. This forms alpha helices, beta sheets, beta turns, and loops. Secondary structure. But above this level, the way that multiple of these domains of the secondary structure can form up will lead to alpha-alpha interactions, so 2 helices that are bundling together, beta-alpha-beta structures where you have sheets and alpha helices interacting, Greek keys, helix, loop, helix structures. So here we see a higher level of engagement where the structures formed as part of these secondary structures have interactions of their own to constitute super secondary structure. And when you see that, you know that somebody has just seen, this secondary structure thing has a lot more going on than just one level, so we'll just dice it up. So now we've got secondary and super secondary structure, and above that we have domain structure, folds or modules. These are all different terms that we use to represent these larger level structures that can heal. So all alpha, all beta, and alpha plus beta units of tertiary structure. So now we've reached this more complex structure we call a fold. And finally, tertiary structure and quaternary structure up at the top. So people have really been thinking a lot about how protein structure emerges from simple sequence. And it's not a 4-step process, if that's what you were thinking. It's not the formation of peptide bonds, boom, secondary structure folds, tertiary structure folds, and then quaternary structure folds. It's not like a sequence of a progression in that way. We have what is essentially a fractal process, Have people used that word with you? Fractal processes are unusual in that you see a behavior emerging at a very finely grained level, like, say, the formation of an alpha helix. And the process unfolding in an alpha helix, in turn, sets loose a process at a higher level, where alpha helices, for example, interact. You have the creation of these super secondary structures, and the arrival of these super secondary structures, in turn, lends itself to the development of a fold. The development of the fold, in turn, leads to the formation of things like quaternary structure. So we have a kind of self-assembly process going on. Sometimes we see that there are helpers that come along to aid structures to take on the proper their proper structure. We also see that sometimes some biological molecules can adopt wrong structures. Have people heard of mad cow disease? In the United States, people get really excited about mad cow disease. That is a really awful thing. Mad cow disease develops because of something called a prion, which is a protein that folds the wrong way. And when that protein folds the wrong way, it encourages other molecules to also fold the wrong way. And that leads to this accretion and a dysfunction in the cow in this case. So these folding processes have really big consequences. When they go right, you get proper structure and proper function. When they go wrongly, you don't get it. Another thing that people don't often associate with structure would be cystic fibrosis. Have people heard of cystic fibrosis? Okay, so in this case, a small change in one amino acid within this protein structure, which normally serves as a calcium pump, this one sequence change, an individual amino acid change, causes a misfolding event. Okay? So instead of folding into this nice calcium pump that's going to go sit in a membrane somewhere, it leads to this other structure. It's hugely problematic. So again, misfolding can lead to serious, life-threatening disease. All right, I'm going to move ahead before I jump into 1,000 other stories. All right, now up at the top, I have these green boxes. I'm just going to say, although they don't take a huge amount of screen real estate, they're actually tremendously important. It's the kind of thing I just litter quizzes with all the time. So I would say everyone here should understand the structural dogma of the central dogma of structural biology. You know, we think of the central dogma of molecular biology as DNA can copy itself in the process of replication. It can transcribe to create messenger RNA. Messenger RNA in turn can be translated to produce protein, right? The central dogma of molecular biology. The central dogma of structural biology is different though. It's pretty straightforward. Sequences give rise to structure. I've tried to tell some of that story by showing the 4 levels of structure and the 6 levels of structure diagrams. A process of this self-assembly, typically, leads to a structure that this sequence points the way toward. Having adopted a structure, particular activity becomes possible. Function follows structure. Structure follows sequence. So this is a really common assumption that you're going to find underlying any discussion of structure. I felt like it was really important to say that right up front. So if you know what the structure is, then you can make some sort of argument about function. If somebody is making an argument that some gene carries out some activity, they're going to be in a lot stronger position if they have a structure that's taken on that accelerates that particular activity. So detailed structures are typically not just about an individual protein or an individual polypeptide. They're usually about complexes because most polypeptides take on activity in company with others, and because proteins interact with other things. You know, if you have an enzyme, for example, an enzyme is just a protein that's really good at catalyzing a particular reaction. But that interaction with another kind of molecule or with another protein for that matter, things like proteases, for example, chop other proteins, these interactions are also key. It's not just that this thing forms the scissors. Those scissors have to be able to interact with the thing it's going to cut. Okay, so structures also help us to understand whether sequence variations matter. If you're spending a lot of time studying mutations that arise in tuberculosis. I work with a lot of tuberculosis people, and I spend a lot of time spotting these new sequence variants that appear in tuberculosis circulating in the population. It's a very big deal. But understanding whether that particular sequence change matters goes a whole lot better when you have some structure in which to understand it. If you know that this sequence variant is out on a loop that doesn't appear to be anywhere near an active site, it's harder to make the argument that it is structurally and thus functionally important than if it is right in the active site of an enzyme. Okay? So being able to understand the impact of sequence variability is much better when we have a structure. And of course, I've already mentioned some proteins don't interact with other proteins, but rather with ligands. Like receptors, for example, right? So being able to interact with particular, to understand which drug candidates are likely to interact with a particular mechanism really benefits from molecular modeling. All right, so structures matter. How do we get them? Okay, I'm going to start, though, with a little bit of biochemistry to think about this. All right, so everyone should take 2 hands now and form a little cup like that. And you're going to put one cup formed by one hand with a cup of the other. Right, everyone has that? All right, so I want you now to wiggle the fingers furthest away from you. So I'm just going to say, open, closed, open, closed. There, you've learned Tanford Transition. Well done, everybody. Okay, the Tanford Transition describes a protein that is a transport molecule. And if I recall correctly, this is mostly about retinol. It's a molecule that you could destroy if you don't protect it as it passes through your digestive system. But beta-lactoglobulin has the ability to take in a little bit of retinol and protect it until it reaches the part of the gut where it can be absorbed. This motion that we just used, open, closed, reflects a change in structure in response to pH. Okay, so as you change from a basic climate to an acidic climate, you see a structural change within this and vice versa. Okay, so there are a couple terms that we want to come to grips with on this. I'm going to start by saying I made this graphic using a structure called 1BSY. The protein data bank uses very compact accession numbers, and so 1BSY is a good example of one of these structures, in this case the one shown on screen. I've used a coloration that uses a color with respect to the secondary structural elements. So we have helices here. I think everyone recognizes our alpha helices. Those are shown in kind of, would you say magenta or purple? Magenta? And did you vote for purple? I thought I heard purple as well. Okay, people prefer magenta on that one. Okay, that's cool. And we have, of course, these beta strands that are interacting as well. Those are shown in mustard, would you say? Could we go mustard on that one? We'll go mustard. We also have some loop regions. These have less rigorous structures to them. Those are shown in white. But you'll note that this structure kind of echoes itself, right? If I could slap a mirror right down the middle, you'd probably note quite a reflection between the two, or a rotation at least. So that reflects the fact that there are 2 copies of this polypeptide that form up into their individual structures, and then they interact with each other. 2 copies. So we call this a Homo Dimer, meaning it's 2 polymers, and they're the same polymer, just rotated, basically. So a Homo Dimer means that these 2 polypeptides join together to take on activity. Next, we have a whole lot of beta sheet here. Beta strands are one of these secondary structures that can form, and we see that these beta sheets have a lot of interacting beta strands all together. And you can see that they're kind of, if you have this way up close, you can actually trace your finger around it. Okay, so this one goes up this way, but then it comes back in and laces back that way. That's the characteristic hallmark of an anti-parallel beta strand, right? We've got these backbones that are interacting in opposite directions with beta strands that are adjacent to them in sequence. So maybe this is 25 through 50, and this is 75 through 100 amino acids, and here they're interacting in this way. So anti-parallel beta sheets, anti-parallel strands to form beta sheets. Okay. So one of these sheets is on one side of the pocket, and the other side is on the other. Now, this next figure, it actually looks kind of terrible, I have to say, but I was really excited that I could make it. The RCSB website, which is part of the protein data bank, has a protein comparison tool. People have uploaded 2 different structures in this case for this beta-lactoglobulin at 2 different pH values. Their names are 3BLG and 2BLG. So what I've done now is to say superimpose one structure on, one structure on the other structure. You see, one had it closed and one had it open, and show me how they differ. So what I've done is to show these overlap very, very strongly. You see how you can't really tell whether that's orange or blue, it's all mixed together? These strands superimpose beautifully. But there's one part of the protein that does not superimpose beautifully. It's this crazy little loop here. It's called the EF loop. So I've got some, a red-colored amino acid here. I just told it to select out glutamate number 89, because this one was the amino acid that they found that changed the most between these two structures. And then there are these little yellow bits on it. Now, the little yellow dots here are intended to show the side chain of glutamate in both structures. So we have, in one case, some fingers that have fluttered open. That's this blue version. And we have some parts where the fingers on the end have closed. That's this orange version. And the red, as you can see, has shifted position between those two. In one case, the side chain of the glutamate is pointing out. In the other case, the glutamate is pointing in. So this change represents a gate on that pocket, opening and closing in response to pH. So I hope that this convinces you that a gross structural change, well, it's not a gross structural change, most of the pocket isn't moving a lot, but we see that this little gate on the outside of the pocket is moving in response to pH, which allows this protein in this structure, which changes between two pH states, to function as a protective barrier to protect the retinol. Okay, so let's look at kind of a classic example of structure. This one is harder to get, I think, but it's a very important one. In this case, you remember I said you could put a mirror down the structure and see that there were two copies of the protein. Here, the mirror is actually shown. That's what that green bar is. It's saying this is a structure that's the same on both of these sides. Okay, now, the structure for chymotrypsin, I misspelled chymotrypsin. That is so embarrassing. We'll have to fix that too. All right, so we see that in 1972, the structure for how chymotrypsin works was published. Now, back in the day, it was a little harder to visualize stuff. And so what they've done is just to show the whole protein sequence now from one end to the other. We spot that the first few amino acids are actually detached from the rest in this diagram, but it's bound together at this particular location. Here we've got a whole bunch of a bunch of little W's in circles here. Anyone know what those are? Water interactions, right? Water very frequently has a big role. If you try to emulate what a protein structure looks like without water involved, you're frequently going to get a very artificial looking structure as a result. So these waters are interactions with water molecules that allow this protein structure to form. Today, you can just download a structure for chymotrypsin. I don't think I wrote down the number of the one I used, and pop it into any viewer. Now, the thing that I can't really show you in a static slideshow is how cool this is. Today, rendering a protein structure in 3 dimensions is trivial. All of us have graphic circuitry in our laptops, for this matter, that allow us to just rotate these structures to heart's content. So if you have a protein structure that you're working with a lot for a particular paper, it really behooves you to try to download a protein structure and mess with it a bit. If you are claiming that residue number 50 has a critical role in its activity, you can say, for example, select residue number 50, color it red, and begin to see how its structure, how changing that could potentially affect the function of the thing. Okay, so we see that even back in 1972, we were able to get data that enabled us to solve structures of these proteins. All right. But how does this do its thing? First off, the catalytic triad, as I said, is a classical story about how enzymes can function. What is a protease? Protease. The A's tells us that it is a protein that is an enzyme, right? So a protease is an enzyme that does what? Cuts proteins. Very good. All right. So the action of the catalytic triad represents 3 amino acids, the 3 side chains particularly, that are in the active site of this enzyme. It explains how they function. So this is a story of aspartate, serine, and histidine. All right. So what do we know about aspartate as an amino acid? Aspartic acid, we could call it. It's the same thing, more or less. Is it acidic or basic? It's acidic, right? Acid is in the name, right? Okay, so aspartate in solution conditions is generally acidic, meaning it doesn't have this extra hydrogen on it. So COO negative charge. What do we know about histidine? What's that? Yeah, it's basic. It's fond of taking on protons. So if you just had those two amino acids together, histidine would probably have stolen the proton away, the hydrogen away from aspartate, and that's how they would be very happy. One has a proton that it really loves and wants to nurture, and the other one is quite content to be without that hydrogen. That's great. Okay, what about serine? Serine has something in common with threonine. Does anyone know? OH. It has an alcohol side chain. Okay. So when we have a polypeptide that shows up to interact with this active site, the proximity of these three residues allows some crazy things to happen. Now, you said histidine likes to take on positive charge, and that's true, but it's kind of ambiguous about it. I mean, arginine likes protons a lot. Histidine is kind of, it's happy to take on a proton, but if you come to get your proton back, it's not going to hide it away, right? It's going to give it back to you. So it's kind of this, it's got this sort of switch hitter-like behavior to it. When you have it in proximity to aspartate, though, it makes it much more likely to take on protons. So serine ends up forming a strong reaction to it forms a bond with this new polypeptide that's come in close proximity to it. And the process of doing that causes a lysis of a breakage of the peptide bond. But to do this, we have to shove certain protons around. So having this proton, having a histidine nearby that can take or give protons kind of at will, gives them a lot more flexibility. And having that acid next to it changes the behavior of histidine a fair bit as well. So serine 195, in this case the serine at position 195 within the protein sequence, attacks the peptide carbonyl, which lyses the peptide bond. We have to use some water that's nearby, and so water has this oxygen that's going to displace the serine oxygen to release the digestion products. So this first step, step one, causes the serine to be connected to the substrate protein, the one that's getting chopped. And water has to show up in order to break that bond apart. At that point, both parts of this digested protein diffuse away, and we're able to get our hydrogens back because of the storage that we can do over at the histidine. Probably not as clear as my biochemistry teacher taught it to me, but these three amino acids in close proximity are able to take on a functionality that individually they would not have. The whole, in this case, is greater than the sum of the parts because of the interactions possible among these different amino acids. Okay, so let's spend a brief moment thinking about backbones. When we think about the backbone of a nucleic acid, we usually think about a sugar and a phosphate, and a sugar and a phosphate, and a sugar and a phosphate. And the sugar acts as an attachment point for a nitrogen base. I think people might remember those three pieces of each nucleotide, right? Sugar, phosphate, sugar, phosphate, sugar, phosphate, each sugar connected internally with a nitrogen base. Amino acids are different, of course. No sugar here, no phosphate here. Instead, what we have is this repeating pattern of NCC. So anytime you see a nitrogen in this chain, and a carbonyl in this chain, you can say to yourself, the bond that connects those is a peptide bond. That's a peptide bond right there. So the amino acid itself is right here. Peptide bonds have kind of an odd property about them, that they always tend to show up in this kind of arrangement. You've got a nitrogen with its hydrogen pointing this way, and you've got a carbon with its double bonded oxygen pointing the other way. As a result, those bonds don't rotate a whole lot. They tend to stay in that trans alignment. Everyone remember cis and trans? Okay, so here we have a trans alignment where the hydrogen points this way and the double bonded oxygen points that way. Not a lot of rotation here. Not a lot of rotation here. They're both peptide bonds. Everyone sees that? Okay. We do, however, have a lot of rotation between the nitrogen and the alpha carbon. Okay, at this point, I must ask someone in the room to tell me how I knew that was the alpha carbon. Why do I call that an alpha carbon? Yes?
So because it's in between the two, the N and the C.
Okay, it's in the middle of the amino acid. That's good. What is it connected to? What is uniquely connected to the alpha carbon of an amino acid? The side chain? Your side chain. Okay, great. So R is just going, in this case, is going to represent our side chain. So the alpha carbon and the nitrogen are separated by this bond, and between them, we have the ability to rotate this bond. If you make the toys up with the little molecular models, you can just spin that to your heart's content. It's a lot of fun, actually, to build these things. So we call that the angle that this amino acid has between its nitrogen and its alpha carbon, the phi angle. It could be 180, it could be 0, it could be whatever, right? There's some reasons it wouldn't be 0, but that's okay. So this rotation is one we're going to call phi. And this one, between the alpha carbon and the carbonyl, we're going to call what? Psi. That's called psi. So if you walk near a psychology department, you're quite likely to see the psi symbol. I find it a little easier to remember because I'm a fan of comic book action movies. And let me see, Elektra in the movie Daredevil used psi knives, right? And they look like that. Right, so there you go, psi knives and phi angles. Okay, so this bond can rotate, this bond can rotate, phi and psi. I just want to say then, if you have a protein with 100 amino acids, how many rotatable bonds do you have on the backbone? 100 amino acids, how many rotatable bonds are there? What's that?
Can you take a guess?
What? 200, yeah, because each amino acid has a rotatable phi and a rotatable psi. Great. Now, over time, people have thought a lot about the implications of being able to rotate these bonds. It means that for a given amino acid sequence, you can rotate an awful lot of bits. So how do we get to a structure from that? So let's start with this question of steric distortion. This is, I'm sorry, it's very, very small here. This is a phi angle. A phi angle of 0 means what? It was just on the previous slide. Phi was which side, left or right? It was on the left. So the one between the nitrogen and the alpha carbon, phi angle of 0 leads to this weird situation called steric distortion. Steric distortion. So this could mean that you have like the nitrogen's hydrogen and the alpha carbon and the side chain pointing in exactly the same direction. Why would adopting a 0 angle between those two be bad? Okay, steric hindrance, steric distortion in this case, means that you have multiple chemical groups trying to occupy the same space. It's simply crowded. Right? So if you turn these angles to a position where their chemical groups are fighting for space, that's going to create a situation of steric hindrance, and thus, in biology, we don't see it very often. So to see a phi angle of 0 is just not something that happens very frequently. Okay, now, we do have some structures that are quite common. So we find that if we look across the structures that we've solved for proteins, we find a fair number of times where we have a negative phi angle and a positive psi angle. these tend to be associated with anti-parallel beta sheets. We saw an example of that in the beta-lactoglobulin, right? The chain that goes up this way and back this way, and those chains interact with each other. That's characteristic of an anti-parallel beta sheet. Sometimes we see that we have a slightly negative phi angle and a slightly negative psi angle. That's characteristic of a right-handed alpha helix. We also have left-handed alpha helices that can happen under these circumstances. But generally speaking, we see that the positions adopted by these phi and psi angles are not random. They tend to fall in general patterns. Okay, so this is called a Ramachandran plot, and it shows most typical phi and psi angles for these protein structures. Okay, we're spending a lot of time talking about phi and psi, but it's a really important concept, I think, for everybody to get at this most basic level. How does this amino acid structure lend itself to structure at a higher level? Okay. Empirically, we see a lot of protein structures out there, and we would love to get a structure for every protein sequence deposited into GenBank. That'd be lovely. The reality, however, is that we cannot do that. It is very difficult to experimentally measure the structure adopted by a protein. One of the biggest challenges required for us is to get a crystal. When we talk about X-ray crystallography, our Cadillac method for getting the highest resolution possible structure from a protein, we really need to have a regularly structured crystal formed from each protein. That is a bit of a problem if what you're working on is a very large protein that is partially embedded in a membrane, for example. Something like that is not that anxious to form a crystal. So we've done well with what we can on soluble proteins particularly. And when we use X-ray crystallography with those, we can measure them down to 1 to 3 angstroms. Right? That's a phenomenally great degree of accuracy for a fineness of detail for the protein structures that we get out. But frequently, we run into problems that don't allow us to get these good crystals going. Instead, we need to use some methods that work with proteins that are in solution instead. But these are most useful when you have a very small protein sequence. So for something like this, nuclear magnetic resonance is a pretty good option. But you see that the resolution we achieve is maybe only half as good as what we could achieve with X-rays. So if we could go the X-ray option, that would be lovely. But frequently we end up having to fall back to NMR because we can't get a good crystal for producing that structure. Happily, electron microscopy is also getting ever better, and we find that our ability to get very tight resolutions through electron microscopy is good even when we have a huge complex. So when you see things like whole proteasomes having their structures determined, it's usually by a method like this, something that can handle a really massive structure. Now, I feel that just saying we X-ray it and a structure emerges is a really big misnomer. So I've tried to put in a slide that asks about this. Did everyone spot the joke on the slide? Okay, it's quite all right to make fun of Watson and Crick. We've lionized them all their lives. But in this case, there's some justification to say that Rosalind Franklin, who did a crystal structure of double-stranded DNA, provided all the information needed to determine what the structure was. Watson and Crick had better Tinker Toys available to them, and they repurposed her data without credit, and in fact said some very awful things about Rosalind Crick in the books that emerged from this. There's a lot of bad blood in this thing. But let's remember that Rosalind Franklin did crystal structures that had all the information needed to determine what the structure of DNA was. We shouldn't say Watson and Crick discovered DNA's fine structure without acknowledging that she made a tremendous accomplishment in that direction. Okay, so at the upper left, we have something marked crystal. Everyone sees that? All right, so we've crystallized a bunch of copies of our protein. It is absolutely necessary that you have a regularly structured crystal where the same bond angles and so on are held for every molecule in that little crystal. We then do diffraction data. We pass X-rays through the crystal, and their passage through that structure modifies the X-rays that are passing through it because of this regularity of bond relationships. That creates an electron density map. Interpreting an electron density map, however, is quite difficult. I've included a paper from 2003 about an algorithm called ARP and its cousin WARP that are used for interpreting electron density maps. So to start with this, I think it's key to say that the electron density map gives you some indication where in 3 space some electron dense region is. Now those electron dense regions are the nuclei of atoms. Now do they come with handy little labels saying, I am the nitrogen of the, at the N terminus of serine 286? They do not. Right? It just says, there appears to be an atom here. So this free atom model is at the first step of inference, to be able to say, from this diffraction pattern, we know that atoms must be hither, thither, and yon to cause these X-rays to be diffracted in this way. Given that, we next need to do what they call auto-tracing. This process is designed to find a continuing stream of atoms that describe a chain, in this case, a backbone. We want to be able to reconstruct that this is an NCC, 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 through this 3 space that represents the backbone. And only once we have that can we start to do things like fit side chains. We often will use an iteration as well, that on our first pass we say, this looks like our most promising carbon, this looks like our most promising nitrogen, whatever. And by iterating, we gradually build up a picture of where atoms are and how they relate to each other along the backbone, and then we can add in side chains. That is a very superficial telling, but I hope that it gives you a start to understanding how these things will work. Okay, so I've shown you this slide already when we talked about repositories yesterday, but I want to represent that each additional structure represents a lot more work than each additional sequence in GenBank. How easy is it to generate a new sequence from some bacterium nobody's ever discovered, right? You isolate its genome, shiver it up into fragments, send it off to CPGR, and back comes your sequencing data. Hurrah! More new sequences, right? But to determine a structure of even one new fold is very, very difficult. The number of places where you can conduct an X-ray diffraction experiment is relatively small. The number of people ready to help you do a protein structure determination by NMR is relatively small. So these are very difficult things to do. This is one of the reasons why we look at a curve like this and say, what a magnificent success it is. Because there's no such thing as an automated structure finder for you. You don't just dump a bunch of proteins on someone and say, I'll be back next week for my structures. This doesn't happen. Okay. So let us come now to Levinthal's paradox. Levinthal's paradox. It would not be possible in a physically meaningful time for a protein to reach the native or functional conformation by a random search of the enormously large number of possible structures. Okay, there are quite a few things in that. First off, if your body begins to express a particular gene, from the time that a messenger RNA is first produced at a gene, how much time is required for the completed protein to be ready for use? I'm going to give you 3 options. Seconds, minutes, or hours? Seconds. Okay, so we know that events like replication can happen at, I don't know, kilonucleotides per second, something like that. In the same way, the processes of expression, of transcription, splicing, and translation can all take place within a space of seconds. It's energetically complex, it involves an investment of the cell, but the total amount of time required is not very long. Generally speaking, we do not say, gosh, from the time this protein is translated, it'll be ready maybe in an hour or so, right? From the time it's translated, its ability to adopt a final structure is something that can happen very, very quickly. Leventhal pointed out that this is statistically unlikely. That, let us imagine that we have a 100 amino acid protein. Is that a big one? It's kind of smallish, actually. 100 amino acids is relatively small. So each amino acid has 2 rotatable bonds. And we'll just presume for the moment that the side chains are just static, right? They just sit there and they don't do anything. Not true, but we could just assume that. So let us now say that each bond could be rotated in 10-degree increments. Is that okay? Okay, how many possible rotation positions are there if we consider each 10 degrees to be a change? 360 degrees divided by 10 is 36, okay. So there are 36 different phi angles for each amino acid and 36 different psi angles for each amino acid, and we have 100 amino acids. So the number of possible side chain conformations we can get would be what?
36 times 100.
What was that?
36 times 100.
Not the right operator, but 36 and 100 are both going to come into this, yes.
72 times 100.
It's actually much, much larger than what either of you are saying at the moment. But you've identified that there are 36 different positions. How many different positions are allowed to rotate in this protein?
200.
200. 200, very good. So there are 2 rotatable angles for each amino acid. So that means there are 36 raised to the 200th power. Okay. If you think of just one amino acid, right, you can rotate this one 36 ways, and you can rotate this one 36 ways. So it's 36 times 36 conformations for an individual amino acid. Right. So that's what Leventhal is saying. There are so many different ways any one sequence could form up that it's not possible that the protein just sort of randomly wanders to it. But people have been able to resolve this paradox by this realization. The introduction of a small energetic bias toward the native state leads to decreases in folding rates such that they cannot possibly, so that they can be compared with what we actually see on the bench. That's kind of shocking. But we've already talked about this in like the 3rd slide. You remember when we said that the 4 levels of protein structure could really be thought of as 6 or more? Right. So we see that very small local interactions lead to the formation of secondary structures. Those secondary structures then interact with other secondary structures. So the amino acid sequence is taking a lot of shortcuts such that it doesn't have to explore every possible conformation until it finds the right one. It can simply fold up this little bit and then that little bit, and sooner or later those folded up bits will begin interacting in an energetic way that drives them to a final conformation. All right, so that is not a process that we can emulate so very well in silico when we do it in software, but our proteins do it every day. Okay, so let us start with some of the bioinformatic applications. How are we doing on time? Have I already run through an hour? Almost an hour. Oh my goodness. Okay, well, I do love proteins, so I'm sorry about that. I'll try to stay on target here. Let's start with the process of comparative modeling or threading. If you are interested in a particular protein in some rarely studied organism for structural biology, your protein probably doesn't appear in the protein data bank. That's unfortunate, but it's true. However, it's often the case that a protein you are studying, even if we haven't done a structure on it, adopts a fold that looks an awful lot like some structure that is in the protein data bank. So if you know that the cousin of this sequence from another organism folds up this way, it's likely that this protein you're seeing that bears sequence similarity to this other protein also bears structural similarity to this other protein. So for that, we need to be able to identify structures that are related to the sequence you're interested in. We can align these two sequences together to see where the principal changes are. And then we can try to alter the structure that we receive from Protein Data Bank to reflect those changes. Maybe I got to replace a side chain here, and maybe I have a crick introduced into the sequence because I've got a proline over there. And then we can put some sort of score on it. One of the ways that we do this is by the SASA, the Solvent-Accessible Surface Area. You can score to say, how well does this structure keep all the hydrophobics together and all the hydrophilics on the outside, etc. So you can score these models, and based on those scores, maybe you can try 50 different perturbations here to reflect these changes. You can see which ones score the best and use those as a basis for another round of possible structures. So this is, again, this process of iteration. We used iteration when we were trying to solve the structure from X-ray diffraction data. Here we can use iteration to keep adjusting our model to be more and more like the real thing. Okay. Oh, did I skip past one? Okay, I think I skipped past one slide. Yes, okay. There are a lot of aspects of de novo structure prediction that are a lot harder to explain than this threading or comparative modeling approach. And as a result, I'm going to have to kind of start at a conceptual basis, and we'll try to walk our way through it a bit by bit. The first is that we still use a lot of Levinthallian approaches for trying to figure out protein structures. In other words, we're going to try a lot of random bond angles and a lot of random side chain placements in order to figure out what works and what doesn't work. Maybe we'll try 10,000 different starting models, all random, to figure out which ones are at least on the edge of plausibility, right? So when we use, when we're starting with this, it's worthwhile to remember that thermodynamically, the structures are not random. Thermodynamically, the structures have tried to reach some sort of accessible minimum of free energy. And I already sort of talked about that. Things like hydrophobics. Hydrophobic side chains don't like being out in water. You want them together to make something like a hydrophobic core. So being able to get us in the direction of a native structure frequently means trying to get a low energy structure. We can't measure its nativeness, but we can measure what energy the structure would take on. So Monte Carlo sampling is one of the routes that we get there. We might try a random set of bond angles and evaluate which of those we can immediately pull off the table as not worth considering to find a subset that are more like plausible structures because they have better free energies. Generally speaking, we would like to capitalize on the successes of the last iteration. So imagine that we started with 10,000 completely random bond angles. Most of those are obviously going to be very, very poor. They're not going to have anything like a free energy that would be acceptable. But some subset of those 10,000 structures are something upon which we can build. So we're going to inform our second round of guesses about what that structure should be based on the ones that were most acceptable from the first round. This is something that we call hill climbing. Okay? And then next we have this idea of Rotamer libraries. It is quite likely that we have no structure exactly like the sequence that we're trying to test in this de novo process. But we do have subsections of this sequence that have appeared in other structures in the past. Right? If you've got 10,000 structures in your data bank, this, say, 5 amino acid stretch probably appears somewhere in there. So you can use the prior existence of these short stretches of sequence in something that we call a Rotamer library to sort of pattern the structure we're testing for this structure by by forcing those 5 amino acids that appeared before to take on the same conformation as was observed before. So these libraries, these Rotamer libraries, give us little hints about substructures within these larger sequences. Okay, that is a start, I think, to understanding de novo structure assessment. CASP is, I think, one of the most amazing ongoing competitions in science. The goal is to do a very fair appraisal of which methods out there for structural prediction are actually working. So every year, someone solves a new structure. That new structure is withheld from the public. Withheld. It's not published in all of these PDB databanks and so on. It's withheld. And only the people at CASP know what that structure looks like. They then give the sequence associated with that structure to all the groups that want to participate in guessing what that structure should look like. And each year, tens of groups will send back their responses saying, we feel sure that this protein sequence adopts this structure or that structure. And there are multiple tests that they're going to do, right? So maybe they'll focus on a ligand plus a structure. Maybe they'll focus on a small hydrophobic protein or something like that. They have a variety of little challenges to test people. So they're given the sequence information, but they must tell you, tell the organizers of CASP, what structure relates to them. The one I'm reporting on here was published in 2017. I think there's another one for 2019? I gotta remember, but. So one can ask, are we getting any better at this? Right? This is an incredibly difficult computational challenge. In the 2017 round of CASP, the large majority of methods had switched to the use of machine learning approaches. If you spend any amount of time near computer scientists or near people who talk about big data, unironically, you probably have heard someone talk about how amazing machine learning is. And in many, many respects, a bioinformatics class cannot be complete without some mention of machine learning. I have only one lecture I try to give on how machine learning works, and it is part of the clinical biomarkers class that some of you will end up taking at the end of the year. Well, in August, I guess. But machine learning is one of those phrases that just shows up in all caps every time any bioinformatics conference shows up. Look at what we can accomplish if we use deep learning methods, or if we use support vector machine methods, or if we use whatever. There are all kinds of different approaches. We see that this has happened in spades in the protein structural world. In this case, in the 2017 cases, 36 groups gave results, although 38 registered. This is actually commonplace. You'll frequently find that in community projects like this, more people will enter than will actually give you results in the end of the day. Some people just know their results are not good enough to look good on the competition and will participate but then not give their result. It's embarrassing, but it happens. All right, so what we find is that in the most recent edition, this 2017 edition of CASP, big gains were made in finding contacts between amino acids that are separated by 24 amino acid residues with no available template. So if you remember our friends, the catalytic triad, we had a histidine, a serine, and an aspartate. Those three are not all together in the amino acid sequence of chymotrypsin. They're actually quite far apart from each other. Physically, in the three-dimensional space of the structure, they're very close together. But from a sequence perspective, they're very far apart. So finding these contacting amino acids, even though they're distant from each other in the sequence, is something that's gotten a whole lot better in this most recent year. So conformations adopted by proteins are essential to allowing them to carry out the activities that they do. Protein structures get measured by X-ray crystallography, by nuclear magnetic resonance, and by electron microscopy. All of these methods are appropriate for some context or another. There's, just because X-ray gives the highest resolution doesn't mean that anybody who can't do an X-ray structure has nothing to tell you. You can learn a lot from a good NMR structure. And frequently, the structures that they produce are more relevant to how these proteins, the structures they take on in solution anyway. Computational methods in protein structure are continuing to evolve. That's going to be a standard for a long time. Structures for small proteins can generally be inferred to a very good approximation just from the sequence. We're getting better at the big stuff too. So I think about where this field sits now versus where it was when I was in grad school. And I'm starting to think that it's moving out of the boutique laboratories that do nothing but protein structure all day to people like you and me who might want to use protein structure information. That, I think, is a very, very hopeful sign, because understanding the structure of a protein is everything about understanding its activity.